Hey guys, today I'm going to talk about transcription assays, protein-protein interaction assays, and DNA-protein interaction assays. These assays can be really important because they can tell you exactly what is being transcribed, which genes are being transcribed in a cell, what proteins are interacting with certain DNA sequences, and what proteins are interacting with each other. The first transcription assay I want to talk about we'll call nuclear run-on transcription. And what happens here is you isolate nuclei of a particular cell. You then incubate it with labeled nucleotides. This will continue to transcribe the genes that have already initiated transcription. Typically, once nuclei are isolated, transcription initiation won't occur, but you can be more cautious in ensuring this won't happen if you add heparin, which will block RNA polymerase from binding. This was really interesting to me because I didn't know that heparin could block DNA binding of a polymerase. But it does somewhat make sense because I've heard that heparin is a sugar and it often behaves like DNA and has an affinity for many different polymerases. In fact, polymerases are often purified using heparin chromatography. So after this transcription with labeled RNAs, the RNA is isolated and then a dot plot is performed to identify the transcripts that have been made. A dot plot has oligos that are bound on a surface so that the sample can be flowed over that surface and the labeled transcripts will bind to their corresponding complementary oligo, and that can tell the scientist which oligos have a transcription complement. This is one way of determining genes that are transcribed. You could also do sequencing, which is probably a more expensive method, but it's much more high throughput, where you convert RNA to cDNA and then add adapters and sequence in high throughput. To measure the strength of a promoter to initiate transcription, you can use a reporter assay. The reporter assay has a vector that has a promoter in it, followed by reporter gene. And that gene is going to transcribe and translate into an enzyme that can give some sort of signal or some sort of report back to the researcher. Usually it's a fluorescent or absorbing molecule that is converted by the enzyme. In this assay, the promoter is variable, so you're determining how good that promoter is in producing the reporter gene. The reporter gene could be beta-galactosidase, which produces a blue dye with XGAL. It could be chloramphenicol acetyltransferase. This will modify a chloramphenicol antibiotic that you can detect using TLC or some other method. Another gene you could use for reporting is luciferase, which will actually produce a visible light with the correct substrates. You essentially transform vectors with different promoters and with the same reporter gene into different cells, and then you can monitor how much of that gene was expressed, and that will tell you how strong the promoter is. You could also measure changes that occur during translation by making changes to the ribosomal binding site. The problem is you can't really distinguish whether or not the changes occurred in translation or the transcription of the gene. Measuring protein-protein interactions is really important in biology to understand how proteins interact and to identify binding partners for protein. They help you understand cellular processes and the function of certain proteins. Immunoprecipitation is a really useful way in determining protein-protein interactions. And I actually misunderstood this method when I learned it for the first time in grad school. The idea is that you're able to precipitate a desired protein and quantitate it by conjugating that protein to a bead. And that bead you can spin down and isolate. To attach that desired protein to the bead, you need a primary antibody that detects the protein of interest, and that's probably the key part of the assay. Then you have a secondary antibody that will bind to the constant region of the antibody, and it could be protein A. That secondary antibody or protein A is conjugated to a bead, and so that's how the protein of interest is isolated. When I first learned about this, I thought that the secondary antibody somehow precipitated the target of interest on its own, how that would occur, but it's actually the bead that brings out that protein of interest out of solution. Chromatin immunoprecipitation is very similar, and it is used for detecting protein DNA interactions. What you do here is you link the protein to the DNA using formaldehyde. You shear the DNA by sonication, and then you precipitate the protein of interest by using immunoprecipitation. You can then do a heat treatment to reverse the binding of formaldehyde. The formaldehyde-induced linkage between the protein and the DNA. And then the isolated DNA can then undergo PCR to identify the sequence of the DNA that was bound by the protein of interest. One trick to isolate protein-DNA complex is to use nitrocellulose. Nitrocellulose doesn't bind double-stranded DNA, but it does bind a complex protein and DNA, and it'll bind protein as well. It also has affinity for a single-stranded DNA. Under certain conditions. On other conditions, you can actually bring out the protein DNA complex and the double-stranded DNA based on the polarity of the solvent you use and the ionic strength. The last technique I want to share with you is DNA footprinting. So for this technique, you get an agent, DNase, or you can also use dimethyl sulfate to cleave DNA. And this method is used to identify the sequence that a protein is bound to. So what you do is you have the protein-bound DNA and you incubate it with DNase or DMS, and you want to get about 1 cleavage per strand of DNA. And that protein will actually protect the DNA that it is bound to, so you won't get any cleavage in that region. And so if you do this incubation and you run the mixture on a gel, you'll get a lot of fragments that correspond to different sizes of that DNA, but you won't get fragments due to cleavage where the protein was bound. And that kind of missing section that you'll see in the agarose gel is basically the sequence that your protein is bound to. DMS is able to get a more precise binding sequence because it's able to cleave closer to that protein than the DNase enzyme would. Dimethyl sulfate will methylate bases, and then after treatment with piperidine, the base will be removed to form an abasic site. And then piperidine will also allow for the cleavage at that site. Another way to break strands of DNA for this assay would be to use hydroxyl radical footprinting. Here you use organometallics with iron or copper to generate radicals that can break DNA strands. I enjoyed getting into some useful assays this week centered on transcription, protein-protein interactions, and protein-DNA interactions. I hope you enjoyed learning about these as well. Have a good one.